From University of Vienna. Actually, he's been traveling around the U.S. A fair amount recently. He was just recently in San Francisco presenting at an FFF conference, and he also took the opportunity to drive down the coast and see some of our beautiful scenery coming down. I think Highway 1 was closed where you wanted to go through, though. That was a little unfortunate, but so you got some good vistas on the way, and we're the benefactors of that trip, and that you were willing to come here and share your expertise with us. So I met Frank actually last summer in South Carolina, and he was leading a CTAC working group on how do we find nanomaterials in complex matrices, and this is his expertise. And we all know this is a really tough nut to crack. How do we find nanomaterials, engineered nanomaterials in soils and sediments and waters, and then try to distinguish those from natural materials that are the same size, maybe similar chemistry, or dissolved materials. And he has a perfect background for being the expert. Well, currently he's in environmental pollution, site remediation, and lectures since 2005, and he leads the Nanogeoscience Division and Nanoparticle Analysis Laboratory in the Department of Environmental Geoscience at the University of Vienna. And his PhD is in environmental nanoparticle analysis. He has a PhD in nanoparticle analysis, so we'll learn a lot from him today. And he's like a lot of students these days, but he was definitely ahead of the game. His background is in chemistry and ecology, so also a really wonderful background at master's level. And his PhD was from University of Technology in Hamburg, and his master's in chemistry and ecology from University of Lunenburg. Okay, and so today, very grateful for you to be here. Frank will be talking to us about nanoparticle behavior, environmental and test media, conclusions from static and dynamic stability testing. Thank you, Frank.
Yeah, thanks, Trish, for the very complete and comprehensive introduction of me. That was really nice to hear what I've all done. The point is that I'm so happy to be here, to see this huge group and visiting that other center on the other side of the United States. I've been to Duke last week or 2 weeks ago, and so to discuss with all of you, and already had a lot of nice discussions before this talk here. I will show you something about nanoparticle analysis also at the end of that talk, but the majority of this, what I will show you today, was influenced or started by the OECD sponsorship program. The question of how can we actually test or investigate nanoparticles, engineered nanoparticles for their environmental behavior. Is there any way how we can find out from the material on our bench how that will probably behave in a natural aquatic system? This is of course important because these particles one day will enter the aquatic system, and we know quite a lot already about the behavior of natural colloids or natural nanoparticles in aquatic media. These can be seen here on that small photo, and so we now have to relate our knowledge that we have about the natural processes in the environment to our engineered particles. First of all, I would like to give you some introduction about our group at the University of Vienna in Austria. Being here for the first time in Santa Barbara, I have realized that we have something in common, even though I have not so much detailed knowledge about the group and the university, but this is actually, as Vienna, a place where you would like to make your vacations. It's so beautiful, so nice. And our group at the University of Vienna is the largest German-speaking university and also the oldest German-speaking university in Europe. It was founded in 1365, so quite a while ago, and it has currently about 85,000 students. So it is quite huge, and it's quite old. Or I would say it isn't old, it has a long tradition. Our group in the Department of Environmental Geosciences is located in the Center for Earth Sciences, and we are featuring 5 subgroups here that are caring for pollutant dynamics, so inorganic, organic contaminants, how do they behave, how are they transported, and how can we remediate them. Then the hydrogeology that is caring for geological processes for groundwater transport, groundwater behavior, and so on. The biogeochemistry and isotope geochemistry is led by Professor Stefan Kremer. He was quite a while at Caltech here in California and moved then to ETH Zurich and then further to Vienna. And my subject here is my group, the Nanogeosciences, and the Laboratory for Nanoparticle Analysis in the Environment. We are currently about 30 researchers and PhD students and technicians in our department. We are still heavily growing on our new acquired projects, so we are looking forward to five more people working in our group in the future. Our topics are not only engineered nanoparticles. We started, or actually I started 15 years ago on natural nanoparticles, the natural colloids these days, and the transport of contaminants of natural colloids in the groundwater, in surface water, in pore waters, and of course also the analysis of these quite complex colloids in natural systems. That's iron oxides, clays, you probably know them. And currently we are starting now for about 5-6 years. We are also working in engineered nanoparticles. And this is about 50-50% of the personnel that is looking into environmental and engineered nanoparticles. So we are also not only interested into the characterization and analysis, we are also interested in the environmental behavior and processes that are taking place on the nanoscale. So how do natural or engineered nanoparticles inflict or interact with a biological media? How do they interact with natural surfaces? How do they change natural processes? We have some projects acquired here, and currently the work is going on in behavior and fate of engineered nanoparticles and titanium dioxide and gold. This is something I will show you today. We are working on the remediation with zero-valent iron nanoparticles, where we want to design the transport properties of the particles. We are also working on the colloidal transport and co-transport of natural nanoparticles, where we are using a lot of field flow fractionation analysis to distinguish or to differentiate the binding of trace and main elements to different size classes of nanoparticles. And of course, we are also looking in abiotic and biotic redox processes induced, for example, by UV light that is destructing or decomposing a natural organic matter, and that is then followed by a redistribution of trace elements between the remaining parts in the surface waters. Okay, so I have structured my talk today into 4 parts. I will first show you some results about the dynamic stability testing of functionalized gold nanoparticles. And after this, I will show you the main part of the work. This is static stability testing of titanium dioxide particles, and the majority of the OECD materials that have been selected for representative titanium dioxide nanoparticles. I will then show you how these synthetic results that we're producing on the bench relate to real natural processes that are taking place in real surface waters. And I will then make a small extension towards the possibility of heteroaggregation processes and how we probably can use, for example, FFF to investigate these heteroaggregation processes with natural nanoparticles. But first, let's have a look to results from a group from Switzerland, from Zurich, and the EMPA at the EMPA in Zurich. They actually did a network analysis here of material flow modeling of titanium dioxide nanoparticles. They also did it on silver and carbon nanotubes for Switzerland. And what they did, actually, based on nearly no knowledge about the real flows of these materials, they just used different compartments like the production manufacturing of titanium dioxide particles, the waste incineration plants, sewage treatment plants, surface water, soil, landfill, atmosphere, and groundwater, and the sediments. And we have an export out of the boundaries of our model and an import, so an import into Switzerland and export out of Switzerland. And then they try to estimate the flows and the storage volumes in, in these different compartments. And when we look at this, we can see here that we have about, in this case, 716,000 tons going to 760 tons going to sewage treatment plant, while in the sewage treatment plant we have some losses through the sewage treatment plant to surface waters, and of course here also a lot of is going into waste incineration plants, because in Switzerland 100% of the biosolids produced in the sewage treatment plant are incinerated. Nothing is used in agriculture, so the flow from the sewage treatment plant to soil is zero in this case. Nice thing, but I have modified this graph a little bit. The real graph looks a little bit different. The real graph has these ranges here. Not only single numbers, but ranges of flow. So the flow of material here can range between 10 tons and 700 tons. Or it can range between 0.6 tons and 45 tons. What does that tell us? That tells us that we actually don't know anything. So the variability of the results is so large, so huge, that we are not sure what actually happens. And that also means that we have to look into detail what is actually happening in these compartments. What are the transfer factors of these materials in these compartments? How are these materials actually acting and behaving in a sewage treatment plant? And when we are looking at the different maximum flows that we have here, then we can see that sewage treatment plants, the untreated overflow water from stormwater release, the reactions in the surface waters, the soils, are the most important compartments. And we need to know how the particles react and behave in these compartments to make these numbers that we have here in the flow analysis more safe, more secure, that we actually really know what amounts of particles are in which way distributed in our environment. So how can we do that? Actually, we know how particles interact. They are aggregating. When we're looking at these this is here the intersection of the Blackwater River and the Amazon. This is the behavior in the salt gradient. We know the particles are simply aggregating here in the salt gradient, in the mixing zone, in the estuary. Most of the particles are captured in our wastewater treatment plants, but they may also be in a high amount emitted into surface waters by these kind of stormwater events. We also would like to know how the particles behave when we are injecting them probably into the groundwater, and we would like to know what happens when we are using these particles and mixing them with standard test media for our ecotoxicological tests. The point here is that this is all mainly physical chemistry, that's color and surface chemistry, and we know that it's just a barrier, a small barrier that keeps these particles apart, and this barrier, this probably electrostatic this energy barrier coming from electrostatic forces or from steric forces, that can be modified by the water chemistry. So the water chemistry controls how these particles interact. So how can we investigate these reactions? We have done, for example, the particle aggregation and deposition experiments. That is not only done in our group, a lot of groups in the world have now done deposition and aggregation experiments, settling experiments on nanoparticles. And how can this be done? We can do this mainly in a dynamic experiment or a static experiment. We can do it in natural waters, and we can do it in synthetic waters. These different approaches have different pros and cons. The dynamic experiment would be you're taking your particle, you are mixing it towards a certain hydrochemistry, so you're putting some calcium in, you're setting the pH, and then you're observing the aggregation of the particles, mainly by using, for example, dynamic light scattering, measuring the evolvement of particle size over time. That would be a classical dynamic experiment. A static experiment is not so well known. It's mainly used in industry, where the particles are mixed as a stable dispersion, and they are put into a certain condition of pH and ionic strength, and then it's been put on the bench, and they let it aggregate and settle over a certain time. And then they look how fast it aggregates and settles by just taking a small sample from top of the supernatant and analyzing the remaining concentration of the particles after a certain time. This is a stability experiment. Done in industry 100,000 of times for new products. We can do these experiments in natural waters or in synthetic waters. When we are doing these experiments in natural waters, fine, perfect, great. We are measuring the reaction of the particle in exactly that water. Santa Barbara Bay or a lagoon or whatsoever. It's a great thing, but it tells me nothing how it will probably react in the Hudson River. I cannot directly transfer the resource to any other condition. While when we're using synthetic waters, we can transfer, we can explain a lot, we can interpret processes in there. How is the calcium concentration in the water really reacting on the aggregation or on the stability? But this is not really realistic because the natural water is a composition of a lot of different species. But when we are starting to use synthetic waters and we are using a wide variety of conditions, then probably we would be able to say a little bit about also complex natural waters. And this is the story I will tell you today. First of all, have a look to dynamic studies. This is a study by Arturo that I really love to see when that came out in the ESMC last year. Where a lot of different natural waters were mixed with a classical titanium dioxide, zinc oxide, cerium oxide particles, and then the sedimentation of these particles was observed in terms of a decrease of dispersed particle concentration here over time. And it's very nicely to see that the influencing factors here are mainly that these stable particles here that do not aggregate and settle, these are conditions of low ionic strength and medium to high organic matter concentration. So the organic matter is stabilizing the particles and we have a low ionic strength, what is actually not screening the surface charge, and therefore it's keeping the particles stable in dispersion. And these ones here that are fast aggregating, that are influenced by high calcium concentration, high sulfide concentration, and relatively low TOC or NOM concentrations. A very nice study, but the drawback of this for us now was that this was true for all these different waters, but the exact influence of the sulfate or the calcium we couldn't really get out of this data. So these results are extremely highly realistic, and we need them. But we can't transfer this now to our Austrian rivers, to Rhine or Danube. So what we thought was we were interested in doing also dynamic studies like aggregation rate constant determination, and we wanted to investigate the aggregation in synthetic waters of different gold nanoparticles. And wanted to find out how the coding actually of these gold nanoparticles would influence the aggregation. And we used 30 nanometer citrate coated, 100 nanometer citrate coated, and 30 nanometer mercaptan undecanoic acid coated particles. This is the citrate molecule on the particles. This is the mercaptan undecanoic acid. We expect some steric or electrostatic stabilization of this particle, of this coating material here on the particle. This is a nice SEM picture of these gold particles. Okay, so what do we do in these dynamic stability tests? We are observing the particle size evolution over time for different hydrochemical conditions. These are different conditions here, and we have a stable condition here, that's the particle size over the time, and we have aggregation starting at these conditions here. We are taking the first part of this aggregation curve here, and investigating the aggregation rate in terms of nanometer per time. This actually, because we are looking at the first minutes of the aggregation here, is more or less a doublet formation rate, so the formation of the first doublet particles. And from this we can determine the aggregation rate constant, what is proportional, what is the proportional constant between the increase of size over time and the concentration of the particles, and we can also extract the attachment efficiency, what is actually the factor, a factor that describes the proportion of the actual measured aggregation rate constant over the maximum achievable, the fast aggregation ratio. Okay, so this is just a slide now to remind people of using the right concentration terms here. This is the aggregation rate measured for different particle sizes as a function of nanoparticle mass concentration in milligram per liter. And we can see that the 100 nanometer citric gold particles are much slower aggregating than the 30 nanometer gold particles. And the 30 nanometer gold particles are, in these highly destabilized conditions, both aggregating at the same speed, more or less. So the problem here is that we are looking into a reaction. and the reactions between two particles. Using a mass concentration here, what directly transfers to a different number of particles present if the particles have different sizes. So under these conditions, the 100 nanometer particles at same concentration are in a much lesser abundance in terms of numbers in the system than the 30 nanometer particles. So of course they are reacting slower, because the C here is not a mass concentration, it's a number concentration parameter. And this is a big problem that we are seeing also in toxicological tests nowadays, that the dose matrix is usually based on mass concentrations. But if the single particle is doing something, we need to convert this at least to number concentrations to get a correct dose response curve here. And if we are doing this and transferring it to number concentrations, we are ending up here in a very nice linear relationship between the number concentration and the aggregation rate. Everything is fine, perfect. So we are on the right side. We are then testing different sodium chloride concentrations towards the attachment efficiency. The attachment efficiency is 1 here at the very high stabilized conditions, and it's 0 here when the particles are stable. And we are determining from this at an attachment efficiency of 1, we are determining the critical coagulation concentration for sodium. For the citrate stabilized 30 and 100 nanometers, and for the mercaptor and the decanoic 30 nanometer particles. So we can see here very simply that the CCC of the undecanoic acid-coated particles is more stable than is higher, and the particles are less influenced by the sodium chloride concentration here than the citrate stabilized ones. That is something we expected. We expected that the undecanoic acid stabilized particles would have an electrosteric effect, so the size of the molecules would work like an airbag between the particles and preventing aggregation of the particles more efficiently than the citric molecules. There was another effect we wanted to study, of course, was the pH effect on the aggregation, and that was very simple. The undecanoic acid and the citric acid have very well defined carboxylic groups. Here, these carboxylic groups have certain pKa's, and when the pH is crossing that pKa value and the groups here are protonated, the particles are losing charge and aggregating immediately. So as soon as the pH goes below the pKa, we have a strong aggregation here. And this is just because all the surface functionalities on the surface of the particles are identical. It's just the carboxylic groups and the pKa's of the undecanoic acid and the citric acid. So simple. Life can be so great. So fine when we have that, but when we are moving further to examples, for example, to the metal oxide particles, cerium oxide, titanium dioxide, we have a distribution of pKa's on the surface. At least for titanium dioxide, we have at least 2 pKa's assumed, but still it's a distribution of acidities on the surface of these particles. So, looking into the NOM interaction with these 2 different coatings, I'm just showing you the undecanoic 30 nanometer and the citrate 30 nanometer particle. It's quite interesting to see that the citrate particle has a reduction of the aggregation rate constant with addition of NOM. So we are doing these experiments a little bit different than others. We are first going into highly destabilized conditions and dosing high sodium chloride concentration. We are setting it exactly to the CCC here. And then we are adding NOM and having a constant salinity background here, ionic strength. We are working in quite constant conditions and still see here with a high NOM concentration from 10 milligram per liter, the reduction of the aggregation. For the undecanoic acid here, there's nothing more or less. So the addition of NOM, even towards high concentrations here, doesn't reduce the aggregation of the undecanoic acid significantly. We think that the reason for this is simply that the NOM can replace the citric acid that is only electrostatically bound, while the covalently bound mercaptor undecanoic acid cannot be replaced by the NOM here, and NOM can mainly build a kind of shell around this functionalization, but doesn't really help to reduce the aggregation of the system. This was also found by Stankus et al. Just recently, that when we have specifically coated gold particles, even with different, it was PVP, citrate, tannic acid here. And we are looking at the influence of Savannah River humic acid in that case, only at 5 milligram per liter TOC. We are even in these cases seeing an increase of the aggregation rate. So addition of NOM to these kind of systems is increasing the aggregation. So NOM is not always unidirectional. It's not always the case that you're adding NOM to your system, you're getting more stable and better dispersed particles. Depending on the coating and the interaction of the coating with the NOM, you even may reach stronger aggregation. Nice study. We got a lot of interesting output. We got rate constants out of that. These rate constants can be used in VLBO modeling, so we can use these fully quantitative data to produce models. Problem here is that it's only very few conditions tested. Very few conditions, some sodium chloride concentrations, some pH, not much in total. So the parameter set is not very wide. Our approach then was, okay, when these static stability, when these dynamic stability studies are so tedious that we only can test always for 1, 2, 3, 4 different conditions, and then the PhD is mainly over, then we have to do something else. So we need something that is really simple and fast and can test a lot of different conditions in a go, can be automated to also to improve the reproducibility. So the problem will be that we don't get really quantitative data. We get limited process information, but we can probably identify master variables. Now what we did, we thought, let's do the stability testing in a way of a matrix. So we are doing a lot of single vials that we are filling with a stable dispersion. We are setting hydrochemical parameters like pH and concentration of an electrolyte. Then we are letting this settle for a certain time. And then we are taking, from a defined depth, we are taking the supernatant, analyzing the supernatant. And we are doing this for many, many different electrolytes that are important. And we are doing this for many different concentrations and many different pHs. And then we are measuring the concentration of the particles in the supernatant, the size of the particles, and the zeta potential. And we are putting this in these kind of plots here. So, and which kind of ions would we like to test? Of course, sodium is important in the environment, calcium is important, but we also need some anions. Chloride would be something, and sulfate here is used not only because it's quite frequently found in the environment, it's also to have a bivalent anion here available. We use this on a background of sodium bicarbonate of 3 millimolar to stabilize the pH, and we use Savannah River NOM as a surrogate for NOM here. We used combinations of a 1-to-1 salt, a 2-to-1 salt, a 1-to-2 salt, and a 2-to-2 salt, and of course NOM. The pH range we tested were between 4 and 8, and the data points in one of these plots here range between 48 single measurements in here up to 63 single measurements in this one matrix. The way how we plotted these was simply in these contour plots where actually the remaining concentration was given here as bright shades for high concentrations. So high stability is in bright and low stability is in black here. And the numbers are here given in remaining concentration in milligrams per liter. The starting concentration here was 25 milligrams per liter. For the zeta potential, we plotted just the magnitude, because the magnitude is relevant for the stability. So if it's high positive or high negative, then it's bright, and if it's around 0, the isoelectric point, strongly destabilized, then it's black here. So black corresponds also with low concentrations on the other side. This is the way how we plot these figures, actually. Now this is now the complete set for a commercially relevant P25 titanium dioxide nanoparticles from Degussa, produced in several thousand, 10,000 tons a year. And we have the concentration plots up here and the zeta potential plots down here. We have the different salts that we added. These are the concentrations in millimolar, and this is the pH. And it's interesting to see that when we're looking for the sodium chloride plot here, for example, at low concentrations of sodium chloride, we have As for a metal oxide with a variable charge on the surface, we have the influence of the pH, having stable conditions under high pH and stable concentrations under low pH, and destabilized conditions at the isoelectric point of the metal. And this nicely corresponds also with the charge distribution, the zeta potential on the particle. We have negative zeta potential at high pH, positive zeta potential at low pH, and that corresponds here with the stability. When we are coming up to high concentrations of the sodium chloride, we have these conditions vanishing here, so it is no longer dependent on the pH, it is just depending on the overall high sodium chloride condition that makes it all highly unstable, screening the surface charge and aggregating the particles. For the calcium, it is nicely and interesting to see that the calcium shows us a different plot. Again, at low concentrations, we have the influence of the pH, of course, but as soon as we increase the calcium concentration, we see that we have a zone of high stability here at the lower pHs and intermediate calcium concentrations. And this goes accordingly to the zeta potential with elevated positive zeta potentials. What happens here is we have a specific interaction of the calcium with the metal oxide surface. The calcium is adsorbing to the metal oxide surface and increasing the positive surface charge. By increasing the positive surface charge, it's stabilizing the particles. So we have the situation that adding calcium to the system makes the system more stable. That is something that we would not actually expect, but in this case, we are working on positive surface charge. So the counterion in this case is the chloride. So it's a single monovalent counterion that we are looking here with a very low capacity of suppressing the double layer and aggregating the system. When we're looking to sodium sulfate, actually we see that over the pH, we have not much interaction or not much reaction on the pH here in the system, and it is all over the place, quite highly aggregated. This is quite astonishing because when we are looking to the zeta potential value, we see something that indicates that the sulfate is also interacting with the surface. The sulfate is absorbing to the surface and turning the zeta potential over nearly all pH values that we have tested into negative values. And we have strong negative values here, minus 20, minus 30 millivolts. That is not directly translated into any kind of stability of the particle. So that is quite strange in this case. And the point behind this could be that the sulfate, as we know it from natural particles, is usually strongly aggregating by bridging aggregation. So the sulfate, as being a bivalent ion, tends to bridge particles and aggregates them. This is something we know from natural iron oxides. Mixing calcium and sulfate is very interesting in the matrix here because we get a mixture of the calcium chloride and the sodium sulfide matrix up here with a high destabilization all over the place. It's less than 10% is left over in these tests after 15 hours. We only have a slight stabilized region here at slightly elevated calcium sulfide concentrations and low pH. The zeta potential all over the place is low. The zone of nearly isoelectric point is quite broad here, where we have nearly no charge on the surface, and the reason is the calcium sulfate absorption to the surface. When we are looking into another situation, and I told you that we have OECD materials in the OECD sponsorship program, a series of materials was selected for representative testing for their toxicity and testing for their environmental behavior. For the reason here, for these materials here, we have different relevant and high volume production materials. This Evonik P25 is something I showed you already, and then we have three different materials from Sachtleben chemistry. They are used mainly as UV filters, the Hombigat UV100, and these two are used in cosmetics, mainly as UV filters in sunscreens. To get them into sunscreens, these two materials are coated. First of all, they get a coating of aluminum oxide on the surface, and then on the aluminum oxide, to make them better dispersible in cosmetics, they get a hydrophilic or hydrophobic coating. What is actually hydrophilic is a glycerine, and the hydrophobic is a siloxane and polyol on the surface. Okay, these plots here are interaction plots of the NOM with of the particles with NOM. What we see actually is that all over the place we receive, with the interaction of NOM, at concentrations of up to 50 milligrams per liter about highly stabilized conditions, highly negative zeta potential, and that is what we actually would expect. The only exception is the Hombicat UV100 that has very high negative zeta potential, but that doesn't really translate into high concentrations. The only thing I would like to point out is here the concentrations are given here now in percentage of the initial concentration. So this is 30% of the initial, this is 50%, and this is even higher, and it's not really good to see, but that's about 80. Again, the plots now for sodium chloride. We had this before, that we had the different salts in one row only for one material. Now we have one salt, sodium chloride, for all the materials. And just want to go quickly through this because when we are looking to the zeta potential plots, we see that these three here, the P25 and the two surface-coated ones, behave quite similar, but with the Evonik P25 having the isoelectric point at pH 5, and the others are shifted to about pH 7. What is actually the aluminium oxide coating? The aluminium oxide changes the surface chemistry, so we have a shift in the isoelectric point. The Hombicat UV100 behaves completely different, and we still don't know why this Hombicat 100 behaves so different. But it also shows that this testing system that we have developed is able to distinguish these different materials from each other. It becomes interesting more or less in the calcium chloride matrix here, where we see that the Hombicat UV100 again behaves completely different to the others. And what we see here also is that the hydrophobic and the hydrophilic UV titane behave very, very similar to each other. The zeta potential plots are quite similar, and also the concentration plots are quite similar. And they are essentially different to the pure titanium dioxide here. So what we see here is the effect of the coating of the material, but we don't see any difference between the hydrophobic and hydrophilic ones. That's strange. That was really not expected. For the sulfate, again, I would like to point out, first of all, we can distinguish the different materials. Again, the Hombicat UV100 is behaving different. I will come to that soon. And these two are behaving quite similar here again, giving very, very similar plots in the zeta potential especially. The reason for these different things is, first of all, these two are not so much different. They are aluminium oxide coated, and they have an siloxane, a polyol, or a glycerin on the surface. These are just attached to the surfaces by sorption. And when you are putting this into the aqueous dispersion that we have here now, we are diluting them extremely. The starting concentration is 25 ppm. That's completely different to what you would do when you're manufacturing your cosmetics. You're using in 10% and more. So we have a very high dilution. What happens is simply the polyols and the glycerine are simply desorbing from the surface, and what's left over is the aluminium oxide coating. So what we are testing here are two fairly similar aluminium oxide particles. That's all. And I think the test shows this very nicely. With the Hombicat, the problem is the Hombicat consists out of quite large and aggregated structures. And apart from this, they have very, very small and stable particles. The remaining particles, in contrast to the large clusters, are around 20 nanometers, and they are really stable. What we think is that we are here testing mainly the remaining stable particles that are still in dispersion and that don't aggregate very much, and that the larger clusters that are representing a quite different material in the Hombicat probably are already settled down. So this is heterogeneity of the material, and this here is mainly desorption of the coating. Okay, another point that was interesting to us was what happens now when we are testing more than just one electrolyte? What happens when we have a background of NOM, what is the case for nearly every surface water, that we have natural organic matter in the background? So we used a very common background concentration of 1 milligram per liter of DLC. We are mixing, we use this is the original plot with the NOM, and this is the original plot with the calcium chloride on the P25, and we mix them together. So at a 1 milligram per liter of DLC, we are making the calcium chloride matrix. And what we can see here is that it changes completely. the pH effect is nearly gone. We don't have anymore any specific interaction of the calcium on the surface, and we have a stable conditions, or nearly stable conditions, with about 50% of the starting concentration up to 1 millimole of calcium, which is also a very realistic concentration for surface waters. So we see here very detailed the interaction of an NOM and the calcium chloride. Okay, intermediate conclusions to this very extensive test of these OECD materials is that, of course, we have to consider a specific interaction of our cations and anions with the surface. They are interacting with the surface and they are changing the surface chemistry. We also saw that the driving force is the pH, but even more, it is the calcium, the NOM, and the sulfate in the system. So what we have found actually is or more or less the master parameters that we have to look into our systems are the NOM and the calcium and the sulfate. When they are present, they may rule the stability of our systems. So also the most appropriate test matrices to explain natural conditions are the calcium sulfate matrix and the calcium chloride NOM matrix, or in calcium sulfate NOM matrix. There's still to be done here. And also the coated titanium dioxide particles that should behave different behave very similar because they're in a condition, in a situation where they actually were not supposed to be at very high concentration, at very low concentrations and high dispersion ratios. Okay, we could not say so much up to now if this is at all realistic what we are measuring. So what does that mean for natural waters? For natural waters, first of all, it doesn't mean much because natural waters don't only contain calcium sulfate. So what we wanted to do is we wanted to compare this with natural reactions. So we used a lot of natural waters, or more or less representatives of natural waters, a groundwater, a lake water, a tap water, sorry, that should be a P, a peat bog water, seawater from farms, and also wastewater inflow and outflow from a local wastewater treatment plant, and 3 different test waters from the EPA. We made a nearly complete hydrochemical analysis here, and from the hydrochemical analysis we went into what we know in hydrogeochemistry, so-called stiff diagram, to categorize our waters. So this is a way where we can plot our waters, and from the graphic itself we can see what kind of character the water has. We grouped sodium and the monovalent cations and the divalent cations, and we have here the anions, and then we added the pH and the DOC. From these plots of our different waters, we could easily address them or tell which kind of matrix that we tested these waters would best relate to. And what we see actually is that with only one exception here, the calcium sulfide type or the calcium DOC type was the major matrix. So in principle, I would conclude here that these 2 matrices are the most important to do. All the others were much less important. And then we went into our testing. So we did all the tests with the real waters again. We measured them, and we made these plots about the remaining concentration, the zeta potential, and the size of the remaining P25 particles for the test results in our selected matrix and the real water results in natural waters. And for the concentration, I think this fits here quite well. Also for the zeta potential, we have a quite good fit here along the line. So we are able actually to more or less, when we have the water quality data, and have the real the respective matrix available, we can predict the behavior here in these real waters. Not so good for the size, but that was mainly related to the fact that the size measurement was extremely complicated with the DLS, and not very reliable at a particle size range above 1,000 nanometers here, for example. Okay, so we are able actually to make a bench test. We take this bench test and measure the water quality parameters, the hydrochemistry of our natural water, and then we can relate this to each other and somehow predict how this particle will behave in the natural system. The problem is we used filtered waters. And that means there is no suspended particulate matter in there, because we wanted to see how a multiple and complex hydrochemical parameter would act on the particles. But suspended particulate matter is an extremely efficient scavenger for particles. So we need to find a way how we can do tests including suspended particulate matter. What we thought, first of all, since we would like to do this in a systematic way, we need suspended particulate matter that is stable, that is available, that we can use for our experiments for a longer time. What we have available actually in the lab is these soil nanoparticles here. This is a 4-liter bottle, and this bottle is 5 years old. You see no sediment, nothing. This is all nicely dispersed, stable, natural soil particles. There's a certain production routine how we do that, and then we are reaching these kind of particles. They do not aggregate, as I said. You can use them for the experiments for a long time, and we're using these actually as reference material in our lab for many years now to test our field flow fractionation methods. We thought, okay, we are mixing this with P25, and then we are seeing how the P25 is interacting with these particles. Great. Doesn't work. I'll show you very soon. So we said, okay, because that doesn't work, we are using the gold particles easier. And this is the reason for this is simply there is no gold in there. But when you're looking into these soil nanoparticles, with this is the extraction routine, it's quite complicated. It takes some time, but it works. If you're looking with an FFF, Icp-Ms method to these soil particles here, that's how they look like aggregated on the SEM filter. You see these are relative distributions. This is not the actual concentration. So these are relative size distributions, just that you see how the different elements, aluminium, iron, manganese, and lead here are distributed over the size. The gray shade here is the overall mass distribution of the sample. And what we can see, actually, iron is shifted to the left, so iron is more related to the smaller particle sizes, and manganese is more related to the larger particle size. And what I wanted to show you is this is the titanium trace. So there's titanium in our natural nanoparticles. We will have much trouble to distinguish the natural titanium from the titanium dioxide from our engineered particles that we put in there. So first, we let this aside as long as we don't have a tracer for the engineered titanium to put in there, for example, a stable isotope or something like this, that makes it possible to distinguish it from the natural one. We skip this here, and we started with the gold nanoparticles. Interestingly, this is now the result of the experiment. We have our natural particle distribution here. This is the iron oxide, is the iron trace from the Icp-Ms. This is the particle size, hydrodynamic diameter that the FFF produced for us. And these are two different conditions. One contained no NOM, and one contained 24 ppm of Suwannee River NOM. Not much difference between these two distributions, and we were quite happy with that. The bold line here is always the one with humic acid. Okay, then we added gold nanoparticles to it at a concentration of 1.7 ppm and shook it and stored it for 2 weeks in the refrigerator. And then we measured it again. So this was, first of all, the result from the gold particle alone. This is 30 nanometer citrate coated gold particles just directly from the standard bottle into the FFF. It has nothing to do with the iron distribution here from the natural sample, just that you see where it would be located in the distribution. And then we measured our natural sample. Oops. And what we see was if we have NOM in the system, we have nearly the same response for the gold as in the standard. So there's no loss of the gold when we have NOM in there. And there's also no interaction between the natural soil particles and the gold particles in our system, because then we would also find gold somewhere here. If we have no NOM in the bottle, or just the NOM that comes with the natural soil, what is about 7 ppm, where we have no NOM in here, we have losses of the gold. About 50% is gone. And we find the gold also associated with larger regions here of the natural particles. So in this case, we have interaction, we have loss of the gold, so it's aggregated and gone. And we also have interaction with the stable natural particles here. For the 100 nanometer particles, we did the same, and that was puzzling us a bit. because the NOM curve here is the one that suffers most losses, while the one without NOM here gives us even a better recovery. So the situation here is just vice versa to the 30 nanometer particles. There can be some explanation that the NOM particles, 100 nanometer gold tends to settle. And we have not shaken the bottle before we measured because we wanted to know what is stable in dispersion. So the 100 nanometer gold tends to settle over the time, and that could explain the about 70% losses here from the single standard. And when you have no NOM on there, these particles may interact with lighter clay minerals attached to them, and that is just giving them a higher buoyancy. So they are settling slower, and we are remaining in the end with a higher concentration for those that interact with our soil nanoparticles. But this is just a hypothesis that we will follow on. The message here is that if we are using particles that have some indicator, some tracer on it, a metal that is not present in our natural particles like the gold, or silver would also be possible because silver is also not present in the natural particles. We can easily distinguish them with this kind of technique, and we can do heteroaggregation experiments with that. If it is somehow contained in our background sample, in our background SPN, this one metal, then we will be having enormous trouble to separate them from each other, and that is still not really resolved this problem. So we would need a kind of stain, we would need a kind of stable isotope introduced into these particles that we can make an isotope analysis, or radio labeling, or whatsoever. So this still remains a problematic question when it comes to titanium, what has a high background of about 5 grams per kilogram in the soil, or cerium and other metals, other metal oxides that are well presented in natural samples. But for gold it works. The drawback here is that I've shown you a mixture of a gold nanoparticle with a natural nanoparticle. This natural nanoparticle is the remainder of a long process, processing soil, just a heap of soil, taking this into the lab and extracting nanoparticles out of them to get them stable as possible. So this fraction that is in that bottle here, where's the bottle? There's the bottle. That fraction in that bottle is already the selected, highly stable fraction of soil nanoparticles. It is highly likely that they don't aggregate with anything else. So the result that we see here is not so much surprising, actually, and we have to think about how we actually deal with that to become more and more realistic here. Okay. Finally, wrapping up. The divalent cations play an enormous role, sulfate, calcium, and others. We have to consider that these are the controlling ions that are setting the stability of nanoparticles. We have to consider the specific absorption of these ions to the surfaces and what this does to the surface in terms of surface chemistry changes. The influence of NOM is strong. It even can cancel out the influence of the calcium and the sulfide ions, but it is not always unidirectional, especially if it comes to surface-modified particles. That was not the case for the surface-modified titanium dioxide particles because two things. First of all, we lost probably the organic coating that is currently in analysis, how much of the polyol and glycerin is going off the surface under these conditions. And we were measuring simply aluminum oxide particles, what we think actually. But for the gold nanoparticles that have a specific coating on it, like PVP or this mercaptan-unbecanoic acid or tannic acid, in this case, the NOM is not always unidirectional and may even start aggregation. The zeta potential that we're measuring can, in many cases, nicely explain actually what happens in our system, but is not always related to our concentration resource. The zeta potential can be, but is less correlated to the stability than we expected, especially for the sulfate results that we saw. Heteroaggregation, of course, needs a lot of attention, and we should start now to make up our minds how we can tackle these, especially with which kind of analytical technique we can tackle that, because dynamic light scattering, static light scattering is just off because we have different entities in there, and we can't distinguish them from each other. Heterogeneity of properties is also something that we have to consider, because what we have seen here is that probably if you're doing an aggregation experiment and you're remaining with a highly stable fraction in the end, and you have some sediment, what does that mean? That means probably that you have two different materials in your sample. It's not probably all titanium dioxide, but you have a fraction of titanium dioxide particles that are very homogeneously negatively or positively charged. That means they are very stable. And you have a fraction of titanium dioxide particles that have a different shape or a different surface chemistry so that they are easily aggregating. So when you're then doing your toxicity experiments on that very stable, nice fraction that remains over after 2 weeks of settling, you're probably testing a completely different material. So we should be careful with that. Finally, I would like to thank you for your attention. And this is the group here. I would like all our people in the group that worked on these different results, and funding was provided by different agencies here, and especially the Fraunhofer Society, CFIC, this is an European Chemical Industry Council, and Austrian Ministries. Thank you very much, and looking forward to your questions.
I think a really good point at the very beginning when you were talking about we usually look at mass concentration and the particle concentration and how it's important to look at that. I imagine looking at the initial particle concentration in that flux, or are you looking at a dynamic particle concentration? And how would you look at that for toxicity studies? The initial particle number or the dynamic change in the particle number?
Over the course of the experiment. In the course of the experiment, that would mean that you have also to monitor the particle size in the course of your experiment. What would probably be possible in the supernatant of a cell test or so, but would probably not be possible in an algae test, or not so easy to do in an algae test. So what we did once was we were setting up a similar experiment without the algae to monitor the aggregation behavior in the media itself. But that is also not very realistic, because as soon as you have the algae in there, you have a part. Exactly. So in principle, we should try to consider first of all the initial concentration, if we are able to do that in the right way. And I think number concentration or surface area concentration is a little bit tricky, but would be something that would help here instead of the mass concentration. And improving our approaches, especially when we are checking our tests, our toxicological tests for testing nanoparticles, we should see if we can develop any kind of method to follow the size distribution in the then upcoming aggregation process.
Oh, good question. So, I was wondering about we so it's basically internal concentration, the mathematical representation of the rate because concentration is considered to be a constant, number concentration.
Yeah.
And so, I mean, the implication is with some changing, these are really short-term aggregations. So, the number of concentration of primary particle is really not changing.
No, yeah, the problem is exactly what you say. I have put this just as a slide here after the last one. How you do this experiment, how you set up this kind of matrix experiment. The point is that you would need, we're starting here this is our starting concentration in terms of mass concentration. This is our starting concentration, and we have very stable conditions for certain situations, and we have highly destable conditions. So the concentration in our supernatant will, for stable conditions, be like this, and for unstable conditions like this. And in between, we have a lot of different situations where slightly destabilized, the aggregation is proceeding slowly forward, and this is then represented by this curve here in the middle. The problem is now to find the correct sampling time. When do I sample the supernatant? When I'm sampling after a very short reaction time, I would be able to distinguish these two situations from this one situation, because that has not far enough proceeded here. When I'm using a too late sampling time, I would be able to distinguish the stable from these two situations. But I would not have the correct resolution to really have all situations covered and nicely measured. So I have to find an optimum measurement time, and for this kind to find this time, I need to make dynamic experiments first. So I do 2, 3, 4, 5 dynamic experiments first to find this optimum sample time, and should be between 10 and 20 hours. And to receive that, the starting concentration in terms of number now must be selected correctly. We were working with titanium dioxide nanoparticles that had an average size of about 180 to 200 nanometers in the beginning. So on a certain mass concentration, this is a very small number concentration compared to smaller nanoparticles. So we started here with 25 ppm. We were then doing the same experiments on 30 nanometer gold nanoparticles, much smaller, much higher number at the same concentration. To reach the same kind of reaction speed or aggregation speed, we needed to set the concentration here to 1.5 milligram per liter. So we first have to investigate the correct reaction environment in terms of concentration of the particles. And this is determined by the size of the particles, and then we get a certain number concentration. Because when we want to sample after 15 hours, the number concentration must be the same, because that determines the speed of our aggregation. And when the particle is smaller, we use smaller mass concentration. If the particles become larger, we use larger concentrations.
I was wondering
we made a lot of tests to figure out the right way to do that. And in the end, we used simple turbidity tests for the titanium dioxide. For the gold, we are using Icp-Ms because we are working at very low concentrations. The turbidity would not be sufficient. But using turbidity at a wavelength of 850 nanometers, so in the near infrared, works quite good. So it's a nephelometric turbidity. So we have an 815 nanometer IR diode pointing into our vial, and we are measuring the stray light or the scattered light at 90-degree angle from that. And that works, as we all know. Has some drawbacks because the particle size, the refractive index, is also a factor in the signal here. So when you have a higher refractive index, then you get a higher scattering signal. So you would probably get some errors here. But we're only working on the titanium dioxide here. We are doing a calibration curve, and we are working in highly polydispersed samples. So it would also be a problem if our particles would be 30 nanometers, 100 nanometers, 500 nanometers, because you would get a complete different reading out of a turbidity experiment for these different sizes, because turbidity is also scattering in this case. Light scattering is a function of particle size in this case. But as soon as you have a polydispersed particle size distribution that shifts from 200 nanometers to 800 nanometers back and forth, and it's still polydispersed, it's fine. It works quite well. So it's a turbidity measurement for the concentration. And we are also backing this up with a calculated count weight in the zetasizer, where we calculate the count weight and compare this also as a kind of concentration measurement to the turbidity signal we get, and take the size and the zeta potential out of the zetasizer measurements. So.
Another question is that is there any medical way to distinguish the kind of content from different organisms?
I got that question from somebody from Evonik that are producing these P25 particles, and he said if there's a single method that I could distinguish between the Evonik and the Sachtleben and other companies. So mainly how he could put a label on the particles that is sure that it's P25. That's of course not possible. It is very, very difficult to distinguish titanium dioxide natural from engineered. Shall I show you? Because I'm putting this in here, and let's see if we can find that. Where are you? Come on. I hope that we have got it here somewhere. We did this together with Kelly Plathy from Michael Cello's group, and we were looking into we were looking into the colloidal content or the nanoparticle content in floodplains behind Milltown Dam. This is in Montana, and this is one of the largest Superfund sites in the U.S. More than 100 years of mining and smelting activities is was done at the Clark Fork River here, and EPA decided one day then to remove that dam, and a lot of hundreds of thousands of cubic meters of contaminated sediment had been accumulated behind that dam, and afterwards was spread along the river. So we took some floodplain soils and analyzed particles in there, and with FFF, of course, and light scattering, and also with Icp-Ms. And this is here one example. This is actually a natural brookite particle of about 50 nanometer in diameter. You can see that it's actually an aggregate. When you're looking at the lattices here, that's an aggregate out of even smaller particles that are about 5 nanometers large, and it contains some lead. This is important for us because we wanted to know if the titanium dioxide or the manganese oxides are the ones that are carrying the lead in the system. But these are the natural particles. This is natural titanium dioxide, brookite. It has a clear crystal structure, so that also can be determined. So we have to distinguish our engineered particles from this one. There are some possibilities to do that. One possibility would be all these peaks that you see here. You see it. Please ignore the nickel. That's the net, the grid in the TEM. But we have iron here. We have some zinc. We have some lead, some zirconium, some aluminium. And that means for us that this particle may be brookite, but it's not completely clean. It contains something else. So we would expect that a technical and engineered nanoparticle like P25 would be clean out of these kind of additional elements, trace impurities, because it is produced from chemical clean material, and there's nothing to contaminate this material in the process. But this would not be possible on a kind of bulk analysis, because you have then millions of natural titanium dioxide particles mixed with some thousand engineered titanium dioxide nanoparticles. How do you want to distinguish anything? You can't find any differences because you have all the other elements around. But when you're using one thing, you're using single element, single particle analysis, when you would be able to do this analysis here on every particle you're interested in, going on that and saying, okay, this is now a titanium dioxide, it's anatase, and doesn't carry any impurities. That would be great. And then you go to the next particle, but it is not possible to do that in TEM because it will take you ages. So you have to be able to do this in a quick manner. And this is something that we call single particle analysis on Icp-Ms. So we are using inductively coupled mass spectrometry and diluting a dispersion of our particles so far that we have single particles going into the plasma. And then you get a flash of each particle that is decomposed in the plasma. You probably know that. A lot of problems with this, and one of the biggest problems is the sensitivity, because you have to distinguish this one flash with these some 100 atoms or some 1,000 atoms that are going from the plasma then into the mass spectrometer from the background of noise in your sample. And up to now we are able to go down to about, for gold nanoparticles, down to about 20 nanometers, 18 nanometers of size. Then the particle is too small and you have too few atoms in there to make a distinction to the background noise. So It would probably be possible to determine this one titanium dioxide particle in there because it's 50 nanometers, but we would not be able to see the traces. They are even too low concentrated. So what we need to achieve is a much more sensitive single particle Icp-Ms spectrometer, first of all, that we can also measure the small sizes and the impurities. And this is very, very, very complicated. And we must be able to measure multiple elements per particle, not only the single one. Currently, we are able to set these multi-element spectrometers like an Icp-Ms on one element, and just measuring this one element, getting a flash for each single particle. But we need all the other elements synchronously. And there are instruments available, it's time-of-flight spectrometers, but they are even more insensitive than the available normal quadrupole or sector field Icp-Ms. So there's a huge potential, because when we could do that one day, multi-element at high frequency and high sensitivity, all this is available. We have high sensitivity, we have multi-element possibilities, even in a single particle mode, but only for larger particles. All this is available in single instruments. We have to bring them together somehow. And this is, I would say, 10 years of work and a lot of money, but it makes sense.
Anybody ever looked at the OAT ratios to see if the oxide signature and oxygen is different?
No. But brilliant idea. Yeah, hasn't been done. But we have something like this, we can do it easily. It would be possible to try to do that. It's another question, how do we get the isotope in the spectrometer? So you would be able to do it in IRMS, but in an IRMS you can't decompose the titanium dioxide. In a multi-collector you don't catch the oxygen. And we have a ring-down spectrometer for oxygen isotopes. That works in solution, so we would need to dissolve it beforehand. I have no idea if that works. It's difficult. That would be something highly interesting to look at that. But I currently have no idea how to achieve that. Okay.